So hello and welcome everybody. Thank you for taking the time to join our webinar. We are very excited to present this exclusive session on AI-powered root cause analysis for drug manufacturing and pharmaceutical plants. So today we are very happy to welcome Ms. Ranjita Sengupta. She's our guest speaker with deep expertise in cell and gene therapy, and she's a biotech leader in the Silicon Valley. So Ranjita, welcome. And would you please take the time to introduce yourself?
Yes, thank you, Julie, and welcome to this webinar. So a little brief background. I've been in the biotech industry for almost 20 years now, working with viral vectors for the past 17 years, and I've been in the cell and gene therapy space for almost 7 years. My experience is in viral vector development and manufacturing, CMC strategy. CMC stands for chemistry, manufacturing, and controls, and cell therapy manufacturing. I've published papers, and I was invited in conferences like the Bioprocessing Summit held in Boston last year in 2024. I'm very interested in advancing regulatory strategy in this space, cell and gene therapy, shaping the evolving landscape and driving real-world patient impact through science and innovation. My LinkedIn profile address is given at the bottom, as well as my email address. So please feel free to reach out to me if you have any questions after the webinar.
Great. Thanks again for taking the time to join our presentation. And here's a brief introduction about myself. I'm the founder and CEO of ProSolvr, and I've been working on this for a number of years now. My background is heavily technical. I did my PhD in databases in computer science at Stanford, and now I'm living my dream life of using AI and generative AI even in day-to-day life, like Alexa runs our house lights. I've been in the Silicon Valley IT industry, a veteran now, and 25 plus years of complex development as well as problem solving experience. So I'm very familiar with the idea of solving complex issues. And that's where I got passionate about technology, and especially technology that can help us be more efficient and more productive. That is the power of AI and generative AI today as well. Here's my LinkedIn profile ID. Please feel free to connect with me. And of course, you're very welcome to reach out on email. We are always very happy to hear from you. I'd also like to introduce quickly Ron, who's on the call. He's our customer success lead. And Ron is your go-to guy for any kind of questions, issues, demos that you want to schedule. He's always there. He never sleeps. But he's not an AI agent. So today's agenda, we have a lot of material to cover, but we will try to give you an overview of a very brief overview of what is root cause analysis, why did we even build ProSolvr, what is the purpose of ProSolvr, and then we will invite Ranjita to walk us through the processes and RCA use cases in drug manufacturing, specifically her expertise, which is in her area of expertise, which is cell and gene therapy. Then we also briefly go over some use cases in pharmaceutical plants. How does AI and Gen AI, these are the new tools and technologies, how can they help us with RCA? And then, of course, our product is there ready to help you leverage this power, right? So we give a couple of brief demos. Next step is, of course, to try it. You're welcome to go to our website. Ron has specifically created this page for you. And we have also have to thank Ron for creating an ebook specifically on like a dozen pharmaceutical problems, RC of those pharmaceutical problems, like with the fishbone template and all that. So Ron, if you don't mind, please put the link for this thing on the chat. And
yes,
people can, thank you. People can bookmark it. And on our website as well, we have several RCA templates specifically for pharma. So briefly, what is root cause analysis? The goal is to find all underlying factors that contributed to a problem. Notice the all. It's not just I find the first one and stop. The idea is to do an in-depth investigation of the entire space. And of course, ultimately to fix the root causes, notice the plurals, there may be more than one, and then take corrective and preventive actions, which is goes by the shorthand kappa in the Six Sigma, Lean Six Sigma world, so that we can avoid repeats. Fewer repeat problems, increase quality and reliability, higher customer satisfaction. Root cause analysis is guaranteed to give you all these benefits. Now, there are several common RCA methodologies that people follow. One of them is 5 why. So you basically ask why, and dig deep. You can also do a fishbone Ishikawa analysis, where you can map take a problem and map it into potential categories and subcategories. Fault tree is similar, but the visualization is different. It's represented as a hierarchy, but all of these things can be actually combined together. You can employ 5 why technique in a fishbone analysis to create the fishbone. Same thing for fault tree analysis. And last but not the least, there's also FMEA, which is a failure mode effects analysis technique, where people actually create a document in a certain format where you can discuss the severity and the occurrence and detectability of expected problems. Now, there can always be unexpected problems, but the FMEA is often a live document where you enhance it as you experience new problems. Now, what are the challenges that people face? These, by the way, are not specific to pharma. Ron and I have discussed with probably 300 people in different domains, maybe now 500. I have lost count. And what we have seen, these are universal concerns. Manual learning, there's too much to read. I have to know about all the problem reports, the prior issues that were resolved. What did we do? What did we find? How did we solve them? Vendor guides, troubleshooting articles, and there's so much knowledge on the internet. Overlooked causes. If I'm not really doing my learning, then I'm likely to overlook some of the causes. Then we have to also collaborate on cross-functional issues, right? Collaboration and coordination can prove to be challenging in many scenarios. The last thing is very related to knowledge retention and knowledge analysis. So you need an expert who knows about these problems and how to solve them best in best ways. And when that expert leaves or retires or moves to another project, then basically you the knowledge, which is the asset in the project, is essentially lost, and it takes a lot of time to train up a new person. So that is time consuming. It is very expensive, and then it's this is complicated by the fact that RCAs are complicated. Oftentimes people don't follow frameworks, so it's unsystematic, little bit ad hoc. It can be very tedious, and it needs a lot of training. So here we are trying to address all these challenges with ProSolvr. This is our mission, and we are agnostic to technology. We are agnostic to domains. We have worked with petrochemicals, with automotive, with pharmaceuticals, with food industry. The goal is to solve these challenges, which are universal, with the combined power of visualization, machine learning, and now generative AI. So it is meant for increasing the efficiency and productivity of our RCA process and the kappa handling. It's based firmly on Lean Six Sigma principles. I should mention here, we are also patented in the US. So it was awarded to us in 2022, and we are very happy that we were recognized for our original contribution. With that, I would like to hand it over to Ranjita, who will walk us through the processes, challenges, and some real use cases in cell and gene therapy, drug manufacturing. Over to you, Ranjita.
Thank you, Julie, for the nice introduction. I want to start with just giving a brief introduction to cell and gene therapy. So cell and gene therapy, it refers to a class of advanced therapeutics that approaches that aim to treat patients and cure disease at the cellular or genetic level. And you can divide it into 2 categories, cell therapy as well as gene therapy. Cell therapy is when you take patient cells, modify them, and then reintroduce them back into the patients. And it can be of 2 categories, autologous or allogenic. Autologous is personalized medicine. It's basically when you take the patient cells, modify it, and infuse it back into the patients. And allogenic it's like an off-the-shelf product when you just take cells from a universal donor and then engineer it and infuse it back into patients. And gene therapy is when you deliver genetic material directly into the patients. And viral vector, which I'll be talking about, has been used as a gene therapy. In many cases, there are approved gene therapy drugs in the US. And the impact obviously is to cure difficult diseases, which are basically not cured by the standard pharmaceutical drugs as we know it, like the small molecule drugs. And in the US, about 7 cell therapy, CAR T therapy drugs have been approved to be marketed, 7 of them, and they're all in the cancer oncogenic space, cancer space. But now a lot of companies are also working in the autoimmune disease space. Next, please. So I just wanted to show 2 flow diagrams for manufacturing of cell and gene therapy products. One is viral vector. LVV and GRV stand for lentiviral vector and gamma retroviral vector. So lentiviral vector is not only used as a gene therapy drug, and I believe there is an approved drug in the US using lentiviral vector, but it's also used to genetically engineer your cells for making cell therapy drugs. And today, my main focus is on CAR T cell therapy, which uses both a viral vector as well as patient's cells. So the first one is showing you the manufacturing process of lentiviral vector. And as you can see, I'm not going to go into the details. Feel free to ask me questions after my talk. But basically, it's a multi-step process and it's done over multiple days. And because you're dealing with live cells, any small deviation in any one step, and because it's a very complex process with multiple steps, any small deviation would have a huge impact on the final product quality, and as a result, have an impact on the patient's life. And that is one of the reasons why it is very, very important to have a very systematic, detailed root cause analysis tool to be able to address a problem if it does arise. Now, going to the next slide. Here again is a process flow diagram, and this is showing how an autologous CAR T cell is manufactured. And here you can use lentiviral vectors. So if you look at the second row, the last orange box which says transduction, that is a step when after you manufacture the lentiviral vector, you use that to genetically introduce your gene of interest into your target cell. So in case of manufacturing of autologous CAR T cell, you do use a lentiviral vector. Again, it's a very complex process. Any small deviation in any of one step would have a big impact on the product quality. Next, please. So root cause analysis for manufacturing failures, they have to be very rigorous, multidisciplinary, and it's a regulatory requirement to be fully documented for compliance. And so the goal is to make the manufacturing process more robust, efficient to reduce cost and delays. And again, it's a complex multi-step process, as I mentioned. Also, it can be patient-specific, as I already mentioned, for autologous CAR T therapy, where you take the patient cells and then infuse it back. So basically, you have to individually, it's a personalized medicine where you have to manufacture a batch for each patient, which makes the cost quite prohibitive. And also, usually the each of the batch sizes are pretty small. So, and then, of course, there is a huge regulatory and safety implication, because of which, if there is any manufacturing failure, you need to do this very, very rigorously. Sorry, I just lost it. Hold on. Yeah. Next, please. So today I will be presenting 3 real case scenarios which I myself experienced in my during my tenure in this cell and gene therapy space. And so the first one, the real case scenario I'll be presenting, is about an equipment failure. 2 is there was an environmental contamination, and the third one I'll be presenting is about reagent variability. So the first case was where we experienced significantly low viral vector titer. Now, what is a viral vector titer? It is basically a measure of the vector's functionality or potency. And if it is very low, then you cannot really use it to transduce your T cells to make your final drug product. And this is what we experienced. As a result, this vector batch was deemed unusable. At that time, we did not have a very systematic root cause analysis tool. So it took us about 3 months to resolve the issue. And the root cause was identified as a contamination of an equipment, which was the TFF column. And this was this particular equipment was used in the step where we purify and concentrate the viral vector. It's in the downstream process. So the consequence to the program was there was a regulatory impact. There was a delay in IND filing. And just FYI, IND stands for Investigational New Drug. And you have to file that in the US, you have to file that with the Food and Drug Administration, FDA, to basically get a license to be able to start your clinical trials. And so there was a regulatory impact, operational impact, because multiple batches had to be manufactured for us to be able to identify and troubleshoot the cause. And then, of course, there is a financial impact, impact to business, where the costs go up considerably. So as a result of that, what was the corrective and preventive action we took? Basically, after that, we concluded that we have to not only replace the TFF column lot, which we had, but going forward, we would have to test all of the TFF columns as a criteria for release. Next. The second real use case was to do with an environmental problem, which was a contamination. So here what happened, we manufactured our CAR T drug product. It was in a GMP facility, but when we went to test it for release, we found the batch contaminated, so it had to be rejected. This took about 2 months for us to resolve. And basically, the root cause identified was that there was a contamination in the environment, unfortunately, which got transmitted into our final drug product. So what was the consequence to the program? The consequence was patient impact. There was a treatment delay, so the patient had to wait for a new batch. And here, when we talk about a patient impact, it's actually very big because these patients are really, really sick, and they are refractory to any current, treatments available. So they really have no option. This is the only one, so it does have a huge impact. Financial, again, obviously the manufacturing each batch which is manufacturing, the costs are very high. So one is that the batch fails, and then we had to remanufacture another batch, and then of course an operational impact too, because there was a disruption in the clinical supply planning and batch scheduling. As I mentioned before, for autologous CAR T therapy, it's personalized medicine. For each patient, you have to manufacture one batch taking the patient's cells. So anytime there is a delay, there's a manufacturing failure for one patient, it impacts the patients who are lined up in to get to be manufactured later. The corrective and preventive action here we took was to review and tighten the environmental monitoring program and to enhance our real-time tracking, and also to increase documentation and procedural training. So the lesson we learned from here was environmental contamination can lead to critical delays in patient treatment and costly batch failures. Next, the third case, this was actually more in the preclinical phase early on when we had started the development of this particular program. Here again, we noticed very low vector titers. So obviously the functionality of the vector was very, very poor. And as a result, we could not transduce our T cells. It failed. This took us a very, very long time to resolve. This was very early on, so we really had no tools to do any kind of root cause analysis. And because we were not very systematic or thorough, it took us about 6 months, which is a long, long time. And here we found the problem was with reagent. This is a critical reagent which is used in the manufacture of vector, which is called fetal bovine serum. And though we had bought the same catalog number, so it was what we thought was the exact same product, but what we realized during our investigation that there is a lot of lot-to-lot variability. So if you use one lot which functions well, not necessarily that the next lot would do also. Here again, there was a regulatory impact. There was a delay in the IND submission timeline. Operational impact, we had to manufacture multiple batches for troubleshooting, and then again, of course, a financial impact. And here, the new practice which we implemented was that going forward, all FBS lots which we receive from the manufacturer, we would do testing in-house to qualify the batch. And the lesson learned from here is that reagent lot variability, even with the same catalog number, can critically impact product performance, particularly these for all of the critical reagents. So the key takeaways from here is that the manufacturing problems can impact product quality, clinical continuity, and most importantly, patient safety. So for that, a robust and timely root cause analysis is needed, as well as a structured RCA process, and then also to be proactive and response to RCS process ensures timely identification of the gap and implementation of preventive controls to avoid recurrence. Next.
So coming back to this slide, Ranjita, it seems to me that the issues you faced took weeks, if not months, to resolve. And they had significant impact on, like you said, patient safety, clinical continuity, even on the financial and regulatory aspects. So do you think this area could benefit from new tools and technologies?
Absolutely. It is very, very critical and important because as we have seen that this is a very, very complex process. Any small deviation, in any single step has a huge impact on patient lives. So it is extremely important to ensure patient safety and product quality to meet regulatory expectations, because FDA in the US and EMA in the Europe are very, very strict. We have to be very compliant and demonstrate thorough RCA analysis and documentation of that. And as mentioned, it'll obviously reduce cost and delays. So this would be very important from a business point of view. And it also drives continuous improvement, and this would strengthen not only our processes, but also ensure efficiency.
Right. Okay. Yes, I totally agree with you 100%. So we move on to the next use case. I want to briefly mention pharmaceutical plants are just like any other plants, like petrochemical plants. They have workers in them, and they are dealing with sometimes hazardous chemicals. So this problem was faced by a real company, and employees were exposed to hazardous, highly potent pharmaceutical powders during the manufacturing process. And they got exposed to these toxic chemicals through inhalation and skin absorption. The cause was determined to be inadequate containment, leading to dust and aerosol formation, and the root cause was determined finally to be human and procedural errors. The consequences, of course, were quite serious. Initially caused skin irritation and respiratory problems, but chronic exposure could lead to more serious consequences like organ damage and increased cancer risk. Now, the company did implement safer engineering controls and containment modifications to prevent the release of these powders and to reduce the needs for protective equipment. Lesson learned was that the better handling of these hazardous chemicals would lead to significant reductions in exposure rates and, of course, improve the health and safety of the employees and the plant as a whole. So these are just to give you an idea of the use cases. These are real cases, not imagined or theoretical cases. And now we have jointly, with Ranjita's help, prepared a fishbone analysis, a quick demo that we can show you on a CAR T therapy manufacturing problem. So here is our beautiful fishbone, and I'd like to show it to you live on ProSolvr. So here it is, and I would like to have Ranjita explain how these categories are structured. Ranjita?
Yeah, so the typical categories of root causes for manufacturing failures in cell and gene therapy are raw materials and reagents. And under that, there are a lot of subcategories because we use an enormous amount of different kinds of raw materials and reagents. I'm not going to go into the details, but like, for example, we use serum, which I'd mentioned, the fetal bovine serum. We use cytokines, plasmids, viral vectors. Contamination of these raw materials could be an issue. There's also supplier lot-to-lot variability, as I mentioned. And then another category would be your equipment and facilities. We use bioreactors, incubators, closed systems. So any breach in there could lead to an error. Process execution is very, very important. Obviously, you need very trained personnel, to be able to execute on these processes. Analytical and QC is another very important category where you have sample handling, reference standards, all of that to take care of. And then finally, documentation and systems, which is very, very important in our field to be compliant. So as you can see, in each of these categories, there are subcategories. So it is the root cause analysis for a cell and gene therapy manufacturing process has to be very, very systematic, very detailed. And so it is extremely important to have a good tool like ProSolvr, which will, I think, increase our efficiency by a whole lot and also help us to document it properly to meet compliance.
Yeah, thank you for that endorsement and vote of confidence, Ranjita. I, of course, agree with you. The process is complicated. And I just want to emphasize here that ProSolvr is not a diagramming tool. We are not online PowerPoint or Canva or anything like that. This is a fishbone that is interactive. Yeah, you can alternatively view it as a fault tree analysis, and it has integrated status. You can collaborate with it. It's like Google Docs. For example, I see a head here, and I can click on it. Say Ron has been working on this issue. He's diagnosing raw materials and reagent problems, right? So it's collaborative, interactive, and also you can attach notes. I don't have time here in this webinar, but you're welcome to see a detailed demo. Just contact me or Ron. The important thing, even over the collaboration and the interactiveness of this fishbone, is that it is self-learning. Now, what do we mean by that? Basically means whatever data we enter here is actually going into a knowledge repository to be used for generating RCA insights for any future issues you face. So let's say I am an expert and then I decide to move to another project, then I need to hand it over. All my knowledge is going with me. Instead here, the knowledge is captured in the fishbone. This is like, basically your repository. This is your knowledge repository. It's essentially the collected knowledge of all your problem solving in your plant and even across companies. Quickly, let me just show you what you can do with it. So first question, let's say Ranjita was not here in the picture. We didn't have an expert like her. How do we get this beautiful diagram? Yeah. So you have multiple options. One is you can, of course, read and create it yourself, which is the least efficient way, of course. And then you can, use our template libraries. So this template libraries is the libraries are built into our system. When you use ProSolvr, you will see that we have almost 300 community templates, and you can say, whatever problem you're having, contamination. So wherever you have these things, you can just preview these and then apply them. Then, yeah, RCA of batch rejection. So we can just preview it and say, oh, here it's already ready-made in ProSolvr. It comes out of the box. You don't have to do anything except apply it. When you apply it, it will just copy it over with one click. Yeah. And then last but not the least, I want to show you that there is also integrated generative AI. Okay, so today you can go into ChatGPT and ask it to analyze a problem that you're having. Now, how do you use it in ProSolvr? You'd have to read the ChatGPT output, then create this fishbone, or use some other tool. So here we have built a bridge automatically to the ChatGPT APIs, where you can actually just generate this thing we call out. Does not share any data, any of your private data. None of it is shared, but just the problem statements, and you can have the GPT create the public insights from the public knowledge for you. So here is a quick demo that I'd like to show you that Ron has created for us. So here we have a new problem. We don't know the categories. We have a blank YY. So I'm going to go into this menu and say, get me a template for this problem that I know nothing about. I am a newbie, CAR T therapy manufacturing problem. What are the possible causes? As you see, it's going to take some time because it's calling out to the ChatGPT APIs and generating the insights for us, right? So whatever you enter here is the only thing that is given to ChatGPT. Nothing more is shared, right? Now we get the information and you will see what we do with it. Basically, the text is transformed into this beautiful fishbone. And as part of the generation of this fishbone, we also ask ChatGPT for other insights, such as, what could be the, most common root causes, or what could be the corrective action. So as you see, it's broken it down into these categories that Ranjita mentioned. What could be some corrective, preventive, and investigative actions? Yeah. So we also have a chatbot, which we're not going to have the time to show here, but essentially you can analyze any problem, airbags from helicopter crashes, plane crashes, CAR T therapy. If ChatGPT knows about it and it is gaining knowledge every day, there are really significant insights that you can get from the public knowledge, because someone in the world could or should have experienced the problem that you're having. Okay, so now getting back to our presentation, I'd like to just quickly say that we have a YouTube channel that we have many demos there. We are happy to work with you to create your own specific use case and solve it. Now, very briefly, how can AI ML help? So I mentioned this is a smart fishbone. It's self-learning, right? So AI and machine learning have become quite mainstream these days. The new kid on the block is, of course, generative AI, but it has caused this revolution. Question is, how can it help us with root cause analysis? That's why we are here today. And let's see, what is generative AI, right? So this is a special branch of AI that can interpret and understand human language. Not only that, it can even create original content. So for example, if I give it a big PDF report that was submitted to the FDA, multiple PDF reports, you can give it 1,000 PDF reports and say, give me a summary of the root causes that were discussed in this incident reports, right? So for AI, it will take maybe a minute or two. For us, it would take probably months, right, to get through that kind of data. So it basically can increase efficiency and productivity by orders of magnitude. Like we ourselves used to create these templates ourselves, yeah. It took us like 2 templates per week at the max, because somebody would read the blogs. We are not experts in pharma, for example. We had to review it with somebody like Ranjita, who approves it or says no, improve it. So we had to go maybe one to 2 templates max. Yeah. And now with the help of generative AI, we can do 1 to 2 templates per day. Okay, maybe 10 templates per day. There is no limit, essentially. So it's orders of magnitude increase in efficiency and productivity, cost reduction through automation. Why? Because the tedious and repetitive tasks, like whatever you put in the fishbone, it can generate a report, right? I can say, generate me the history. Who took her? Why did it take 6 months? As a manager, you might have that question. Why did it take me 6 months? What was investigated? So that you can understand, maybe don't do this anymore, right? Don't go down the wrong paths. What was the red herring in my analysis, right? And enhanced decision making. AI, we have to really view it as assistance. It is not going to replace my job, because human decision making is still supreme. And but the virtual assistants can really, really help us, right? So with that, I I would like to actually open it up for Q&A, but just quickly, one or two more slides. Cause suggestions can come from AI, action recommendations, what kind of things you need to do to improve quality. And then these are, again, new kids on the block, generative AI. Can it help me, with the knowledge analysis in English? I want to ask it, tell me the most common root cause. What should I do for this problem? What actions would be the most effective, right? And then also agents, which is the next frontier. Can you have an AI agent that can actually inform you that, hey, your reagents are having a lot of problem? So what makes it an AI agent is that it operates autonomously and can be used to notify or even do surveys, right? So it's very, very powerful, and you can use, of course, our product. There are many demos, as I said. We don't have time to go over this right now. Just to summarize the benefits, first of all, it automates knowledge analysis by orders of magnitude. It will give you the efficiency, right? Reduce your training times, because it's my assistant now. I am a newbie, but it doesn't matter. It has the knowledge that I need. We can solve problems collaboratively. This is like Google Docs. You work together as a team on the same fishbone. Yeah, the fishbone gets updated automatically. Your changes will get merged. Prevent problem recurrence. This is the holy grail. We don't want to see the same problem again, right? Of RCA. Enhance customer satisfaction, achieve operational efficiency and excellence. Yeah, these are the main goals. And in ProSolvr, we live and breathe this mission. This is what motivates us. We would be very happy to work with you, show you a demos, any or many demos. Try our app for free days. You can just free for 30 days. Sorry, app ProSolvr Tech. You can follow our page on LinkedIn. You can subscribe to our YouTube channel. And of course, you're very welcome to contact me, Ron, or our support emails anytime, any questions, any comments. We're always very happy to hear from you.